Hi everyone, welcome to today's webinar, Conducting Vivarium Risk Assessments in the Research Environment. We're glad you could join us. My name's James, I run webinars and events here at Triumvirate Environmental, and I'll be your moderator for today's event. Before I turn things over to our speaker, I just want to run through a few housekeeping items for those that may be new to Triumvirate webinars. Though your microphones are muted, we do want to hear from you, so if you have any questions throughout the webinar for our speaker or any technical issues, please feel free to type them into the questions box, and those are over on the right-hand side of your screen. We will have an open Q&A period after today's presentation. We'll get to as many as we can, and feel free to type them in as they come to mind, and we'll get to them at the end. In the event we don't get to them all, we'll follow up in the coming days, and we'll also share the contact information for our speaker so you can get in touch with him. Finally, keep in mind you will receive a copy of the slide deck and the webinar recording in an email tomorrow, so everything you see today you will get a copy of. Our speaker today is Tony Gemilaro. Tony is an EHS manager and senior consultant for Triumvirate Environmental. He has over 25 years of experience in the biotech and biopharmaceutical industry, as well as the public sector, healthcare, and academic environments. Tony's demonstrated an ability to implement corporate EHS standards and programs, utilizing his knowledge of EHS principles, management systems, hazard prevention and control, to educate and grow awareness. Tony's also been an adjunct faculty member at Anna Maria College, Northeastern University, and the University of Phoenix. He has a BS degree in biology from Boston University and a master's in biological sciences from the University of Massachusetts. And so with that, Tony, I'll turn things over to you.
Thank you, James, and welcome everybody to the webinar here at Triumvirate Environmental. I will talk a little bit about an overview of the regulations, really focusing a lot of vivarium risk assessments as they relate to chemical and biological exposures, but also talk about other points of interest for the group, and certainly looking at what I think concerns about exposures to animal allergens, maybe occupational exposure limits, sampling or biological assessments as approved by institutional biosafety committees and IACUCs. Obviously, those of you on the line are familiar with IACUC, the Institutional Animal Care and Use Committee. So certainly looking at risk assessments through the protocols is very important. There are standard protocols out there, many different versions, looking at not only protecting people, but also protecting animals, and certainly going through requirements under OWA, the Office of Laboratory Safety. So we want to take a look at that as well. So worker and animal protection is very important, and also vivarium management is very important, I think, to a lot of us here. So throughout the course of the presentation, I'll focus on some best practices. I'll end with some case studies and examples of success. But I do want to start off with what I like to call the good, the bad, and the ugly. And so let's take a quick look at that. So you all might be chuckling when you see this, but is this good? Is it bad? Do bovine or horses or cows carry anything that could be potentially infectious or dangerous when handling, along as the other issues associated with large animals? But we're looking and thinking about what are the hazards here? What's the worst thing that could happen? What do I need to do to be prepared when handling animals? And what's the prudent practices, protective facilities, and protective equipment needed to minimize any risk? So this might be an easy one, okay? This one usually gets a lot of people, okay? Would you let your child do this, right? Is the snout of a pig safe? Of course, we don't know where that's been. But we want to also ask those same questions. What's the hazards? What's the worst thing that could happen? Should we encourage this type of behavior outside the lab or inside the lab? So here's another example of the ugly. Don't know if you can see this, but it's probably a young college student doing her first surgery on an animal. Okay, and again, the questions that I have here, what are the hazards, what's the worst thing, comes from the laboratory safety standard, asking about prudent practices, not only with chemicals, but with biologicals and the use of animals. So this brings us to questions about regulations. There are some key regulations that are out there. It is a privilege, in my opinion, to work with animals in research, and there are some common concerns for the care and welfare of these animals. And so some testing falls under the jurisdiction of 3 main government agencies. So we're looking at congressional acts that are out there. So the primary responsibilities for the Animal Welfare Act, AWA, and also looking at OSHA, CDC, and NIH for the use of animal biosafety levels and infectious agents. OSHA, as I said, OSHA fact sheet is out there that deals with basic use of small animals and protection against animal allergens, animal bites, and scratches, and so forth. So we're really looking at some details regarding the care and use in research, and looking at implementing these regulations at our own facilities and then maintaining these facilities. Before I forget, also there are different communities or towns and cities that may have their own regulations. The only one that I really know of is Cambridge. So anyone in the Cambridge area does know that there is an animal inspector. There's a veterinarian that does go to all the animal facilities in the city of Cambridge. If anyone else has that requirement in their city, but certainly would like to know that, maybe at the end of the presentation. And so also as part of the regulations, if you are part of APHIS or through USDA required programs, then you might be subject to an unannounced inspection. Small animals, maybe not if you're just mice, depending on whether or not you're going for accreditation or not. Okay, so here's the basic program management structure. It may be different at your organizations, or maybe you're in the process of developing something or revising a program at your facility. It usually starts with the CEO or the president of the institution or industry, who will appoint in writing an institutional official, or IO. And this IO is the individual who, as a representative of the senior management, will provide the ultimate responsibility for the program, and also provide resources, planning, and ensure that the program is aligned with the institution goals as well as compliance. In some cases, the IO may serve on the IBC, Institutional Biosafety Committee, or on the IRB, Institutional Research Board as well. So you can see here that there are different levels here. So obviously a very properly constituted and functioning IACUC is important, with a chair and various members. And I'll talk about members that would be on that committee. IACUC administration, someone to monitor the program, an AV or a veterinarian, attending veterinarian, who would be in charge of the program, overseeing husbandry, the care and use of the animals, animal housing, and then any facility plans and maintenance that goes with that. And then obviously the PI, principal investigator, and scientists, who will submit project protocols and will conduct the research, the valuable research, as part of the program. So obviously the essential components here involves a properly constituted IACUC, veterinary care, environmental housing, and kind of overall program, including training, which I don't have listed in the slide deck. So training would include a number of things at your institution in terms of the experience level of the PI as well as the handlers. So when we're looking at who's responsible for the program, we're looking at, well, it could be different for anyone or other people at every institution. It's not the same. It might be different at your place, or maybe you're someone that wears many different hats and you have responsibilities for the animal care program, and you may have experience for other programs. In many cases, the EHS person is also involved. My recommendation is that an EHS person sit on the IACUC and assist with maybe overlapping responsibilities and programs with the Institutional Biosafety Committee as well as the IACUC. So certainly, this animal care program means that policies, procedures, standards are put in place to achieve the humane care and use of animals in the laboratory and then throughout the whole institution. So it's not just defined or confined to one location. And so if we were to go to the guide and look at the guide in more detail, we would know more about animal care. So the intention here is just to give you a basic overview, but I do recommend that you look at the guidelines in more detail and learn more about the responsibilities for the IACUC, the attending vet, the staff, and students that might be at your institution. So if we look at the process of risk assessments or the process of reviewing protocols, certainly there are a number of different ways that this can go. If you're ensuring a duly constituted program or IACUC, we're looking at all of the federal regulations in place, appointing people in writing, making sure that all members understand their role on the committee, and that you have kind of a responsibilities to look at the protocols from different eyes or different point of views, depending on the area of specialty. So certainly when you're reviewing protocols, there could be some common hazards that are involved, and I will get into more detail. So you might want to have a separate meeting with the principal investigator, EHS, and staff to review some of the hazards, and then put in place some SOPs, procedures, additional information that will support your IACUC protocols and registration documents. Or you might have some special hazards, maybe biological toxins, botulinum toxin, or certain chemicals that are highly hazardous and toxic that would need some additional review. So as I mentioned, then you might need to bring your review before you start any animal work to the Institutional Biosafety Committee, maybe an initial meeting at the IACUC, bring it to an EHS review, or go back to the PI for additional information. So I hope that's clear. And obviously the veterinarian plays a key role. So what I have found to be effective is submitting the initial protocol to the veterinarian. He or she will provide information and feedback, and then help guiding where that document and review process needs to go next. The other thing that I did not have here under membership is having a community representative. So you need a community rep that will sit here and understand these protocols from the layman's point of view to make sure that people are doing things in the right way. So some of the procedures that are in place, this is not, this is just part of a list. There's certainly many more SOPs, procedures, and controls that you can put into place. It's important to have a main review protocol or SOP that deals with the whole program. And then it's important to really clearly define, as I said earlier, the roles and responsibility. What is the authority of the attending veterinarian? Can he or she shut down operations if there's anything noted that could be cruelty to animals, or if there's a problem with personnel being exposed to certain procedures, or if there's an exposure to isoflurane or any other gases that could pose a risk to people. So also think about, and some of you may already have these policies in place, but what about studies with severe disease, death as an endpoint? Death should not be an endpoint, but you need to review the study in more detail. What can you do to relieve pain and distress? What's your euthanasia procedure for animals? And you need to have experience and training records in place to prove to any inspectors that you have put these in place, and you have people in place who know what they're doing and can effectively administer the program. And then some of the small items would be survival surgeries, blood collection, training people in how to use needles and syringes, and how to take blood properly so that they're not exposed to any sharps as well. So we're coming to a poll question here. So I will read this question. Okay. So there are several actually questions. So James, I probably I don't have the, I guess the A, B, C, and D options here.
Yep, we just launched the poll, so everyone should be able to vote now. It should be up on your screen. The options are animal allergens, exposure to gases, animal bites and scratches, and other ergonomic issues, such as back injuries or repetitive motion. And then, Tony, we can just share those results in a few seconds.
That sounds good. So we'll give you about 15, 20 seconds to do that, and try to think about what are the major risks in your facility what is that burning question or something that would keep you awake at night in terms of the risks that you have at your facility? So James, I'll let you do the timing, and we'll give you just a few more seconds to think about that.
Great. Looks like we got just about everyone, so I'll close that and share the results. Tony, we've got 77% selected animal bites and scratches, 66% animal allergens, and 61% ergonomic issues, and 55% exposure to gases. So keep in mind, there were multiple selections here.
Okay, all right. So it's interesting that animal allergens is out there. I think that is a common issue. How do we protect people from allergens in the work environment? Do we handle that with PPE, engineering controls? Do we use respiratory equipment, and whether that's a voluntary program or mandatory program to prevent allergens? And then certainly ergonomics, I know, has been a question out there to prevent any risks to repetitive motion injuries, shoulder injuries, neck injuries, things like that. So very good. Thanks, James. That's kind of a nice highlight of the group. So I will be talking about some of those areas and risks at your facilities. So when we look at the protocol forms, there's quite a few risks. So I think you've already touched upon some of these, but I would just want to give you a full, what I think is a full list. I may have missed a few things. So it may or may not apply to you, but to keep in mind, my area of specialty is biological safety, but I have other areas of specialty. So biological toxins or agents are on the top of the list. If you're working with infectious agents, cell lines, or select agents and toxins. Chemical agents, so if you're injecting certain carcinogens or you are purchasing some chemicals of interest and then injecting them into animals, what happens to the byproducts of these chemicals? Where does it go? How is it handled? How is it injected? Is it sprayed onto the animal, whether it's chemical or biological, or injected directly into the animal, or are they ingesting it? Okay. I have not seen a lot of radiation work recently, but there are options where people do work with radioactive materials, certain radionuclides, like P32, iodine-125, and then physical hazards, needles and syringes. There's always the potential for needles to be used and possibility of sticking someone accidentally with a needle or while the animal is moving around. I know I've been talking about mice and rats, but there are other animals like rabbits that can scratch you, as well as other larger animals like monkeys and so forth. So these animals can provide kind of a risk to you. Exposure to allergens, so certainly that's a big one, and we're going to talk about the different allergens that come from mice and from rats, and things to be aware of, and those controls to protect you against those potential allergic reactions. Okay. Cleaning chemical agents, it's not directly related, but if you're using certain agents like Spore Cleanse, it's pretty nasty, contains hydrogen peroxide, it also contains acetic acid, and some other agents that could be potentially irritating to the nose and throat and respiratory system. Related to ergonomics, obviously lifting, ladder use, washing down ceilings and walls can be difficult, and that can create wet floors with the potential for slips, trips, and falls. And obviously, the last thing is cage washers and other equipment that could potentially put people at risk, looking at how that's used and if there's any aerosols that are being generated that could also contribute to potential allergen exposure. So definitely identifying these hazards, trying to mitigate the risks is very important. Reduce to minimal levels or acceptable levels. But these assessments too, looking at the risks, is kind of an ongoing process to make sure that you periodically check the program, check individuals working with these different hazards, and then moving forward and reviewing. So one of the things that actually is very important too is making sure that there's a occupational health program in place, looking at the health history, sending out a questionnaire about allergies. People could have allergies existing, could be they have a history of allergies, or it might be an onset where they've never been exposed to animals before. And so being in the workplace, would make it a potential workplace exposure and something that would be recordable on the OSHA log. Briefly, ergonomics is something that we looked at, talked about, lifting, bending, doing anything in an awkward posture or static posture for long periods of time. Cages aren't heavy, but if you're changing a lot of cages over the course of a few hours, that could increase your risk for repetitive motion injury. And certainly the occupational health history would look at work with such agents as tuberculosis, which means people would need to have surveillance. In some cases, if they're working with certain agents, they would need a hepatitis B vaccine or some other vaccine, and then protecting against potential exposure. So vaccines are important. I don't know if anyone out there works with snakes or potential snake venom, but certainly a recommendation is to have that antidote ready just in case something does happen. So the last item here is personal protective equipment. Respirator qualification is important, and it is important to provide training and a medical review to make sure people are fit for wearing respirators. So I didn't do this as a poll question. Maybe it's something you can ask at the end. I don't know if people require respirators or it's voluntary. Many of the places that I work at, it's voluntary. It's not required unless there are individuals that have come up with a risk for an animal allergen. So we want to try to reduce, companies want to try to reduce that risk so that employees can continue working in that location. So I think what's really important here is to determine the severity and seriousness of the risk. So really looking at a matrix here of the likelihood of something happening and the consequence. What would be the worst case? What would happen? Is there a low risk, high risk, and what would happen if somebody was to be exposed? Would they be out of work for a while? Would they be a long recovery? And certainly you'd want to work very closely with your healthcare program, your occupational health physician, to make sure that you can prevent recurrence and other exposures for other people. So it's also important to do periodic inspections and report any potential hazards or even near misses. If there is something you think might be a problem, it's good to nip that in the bud and address it early on. In terms of biological or biohazard risk assessments, the questions for you to think about and maybe take away from this presentation, what are the biohazards or procedures involved with the potential material? Biological material risks are a little bit different than chemical risks. You really have to understand the experiment involving the animals. How is it being handled? What's the worst thing that could happen? As I said, the consequences to personnel and the community. How common are these adverse effects? There are some published laboratory-acquired infections that are out there that have been published over the years. They could be rare, moderate, or frequent. If we're looking at exposure to the number of TB-related cases or other types of exposures from needle sticks, what are they? Are you prepared from an emergency point of view, right? So do you have a disaster plan? What will you do with the animals if there was a potential disaster in the area, chemical exposure outside the building, or a disaster like a main gas line ruptures outside the facility and you have to shut down the facility? How would you care for those animals? What would you do? Okay. Or those of you maybe indirectly, if you're involved in a hurricane situation like they have had down south, what would you do with those animals and what type of surveillance would you have in place? Okay. So the main thing here too, or one of the main things, is how would you control your hazards, control your risks? Would you consider eliminating, substituting with a less hazardous material, adding additional engineering controls such as more ventilation for your work with isoflurane, or additional personal protective equipment? So really that kind of boils down to highly pathogenic work with select agents. You want to take extra precautions, extra regulatory requirements. How do you procure material, maintain it, and then dispose of the biological material that's being removed? Okay. And you may want to consider the people that are working there, if they are, if there is the use of immunodeficient and genetically modified animals. What's the risk of anything being shedding from these animals? that could create some human pathogens. Okay. Certainly these are a few things for you to think about, some food for thought. So agents that are infectious to humans may include blood, plasma, or urine from macaques. Monkeys carry the monkey B virus, herpes virus. There have been some exposures and monkey bites as well, and scratches in the Boston area and other areas in the area. So primates can harbor some viral and bacterial diseases that are infectious to humans. Monkey B obviously can be transmitted from animal to humans via bites and scratches. So that might be a concern. How do we protect people from those exposures? And so we'll come up with some controls in more detail later. But certainly controlling animals, looking at the source of the material and where they're coming from. Many times now animals are bred on separate islands so that they don't come in contact with another monkey that might be infectious. Okay. So there was a question about laboratory animal allergies, so I think I anticipated that. We can't cover everything in this presentation, but allergic reactions to animals is among the most common condition that adversely affects pharmaceutical research workers as well as workers in colleges and universities, institutions, hospital environments. So we want to take a look at this in a little bit more detail. Okay, so some of the animal allergies that could occur would be nasal symptoms. Pretty obvious. You're breathing in some animal dander and dust, sneezing, coughing, creating itchy eyes, maybe some allergic conjunctivitis, rashes that could, as a result of contact with material, urine, bedding, certain surfaces that may come in contact with animals could create a rash. So symptoms usually evolve over a period of 1 to 2 years, then which may lead to an acute anaphylaxis, which could be a severe whole body allergic reaction. Okay, that's only in a small number of patients, but I think as a safety professional or animal care professional, it's important to try to minimize those risks so that the acute anaphylaxis does not occur. In rodents, the primary allergen is a protein or protein produced, which is produced in the liver and excreted in the urine. Oops. So this is the MUS, M1. Okay, it's a urinary protein that could potentially cause the mouse allergen. The rat allergen is known as a human allergen protein in the urine and saliva of the animal as well. Okay. So there are several routes of exposure. There's inhalation of these aerosol allergens. If you're generating any aerosols, that creates a risk. Aerosols from the bedding and aerosols from any biological agents or chemicals. Contaminated bedding might be a more obvious one, or if you're changing the animal cages. Although when we talk about prevention, as I'm thinking here, it might be good to have animal cages that are disposable as opposed to cleaning them, but obviously there's different options and costs there. When looking at exposure, direct skin contact and eye contact might create and increase the risk of allergen exposure. So thinking about these risks and assessing them, 2 questions come to mind. What are the typical exposures? What tasks have the highest risk? And so the second one is important, looking at how people are handling, looking at maybe observing animal handlers and procedures when they perform their daily tasks would be important to look at and discuss. So the activities, as we mentioned already, just to put in a summary, handling animal in cages with bedding without ventilation could put people at risk. Manual cage changing and dumping soiled bedding without proper protection. So you can see here from the picture, there's somebody with a N95 respirator on, gloves, a covering or some kind of lab coat, and the cages are changed in a biological safety cabinet that will contain any potential allergens that might come out into the environment to protect that employee. Okay. Monitoring can be useful. However, this is a controversial issue. There's no OSHA standard method for allergens, but it's good to be vigilant and proactive because these allergens can attach to particles or particulates and can be hazardous when deposited in the respiratory tract. Okay. Inhalable and respiratory fractions includes particulate matter that lead to the airway region and into the nose and mouth. Okay. So I want to try to reduce that. So this brings us to the next poll question. Okay, so are there any liability issues with lab animal research areas, such as animal bites and scratches, which I think we've talked about that already, exposures to chemicals or biological agents? But I think, James, you have the 4 or 5 questions and options for people to check off. So I'd be curious to find out if there's liabilities in terms of students, population, and community.
Yep, and that poll question is now displayed. Keep in mind you can select more than one answer. The options are regulatory reporting, grant violations, employee or student exposures, and other. And if you select other, feel free to type in your response in the questions box and we can share some of those as well. We'll give everyone a few more seconds to respond and push the results.
Sounds good.
Great, so looks like we got just about everyone in. Let's go ahead and share those. So Tony, 98% of our audience selected employee or student exposure, and 78% of the audience regulatory reporting. And then grant violations came in 45% of attendees.
Oh, interesting. So I'm going to start with the grant of 45%. So it sounds like people may have a concern with losing their grant if they don't do the right thing. So I can see where that would be a liability issue. It would certainly affect the funding of that institution's requirement, but also would lead, obviously if you lost your grant, then there could be some safety violations or animal care violations as it relates to the grant renewal or continuation. It's highly unlikely. I've only seen a couple of situations in my lifetime of a grant being revoked or PIs not doing the right thing. So it certainly is important to be vigilant in reviewing these protocols in more detail. So employee exposures is very important, especially students. So from the student perspective, if you're at colleges, it is a prudent practice to have the institution have something in writing, or the student has to sign something that there's maybe a potential for exposure to animal allergens or biological agents. It may depend whether the students are undergraduate or graduate school as well. If they're immunocompromised, then they could be a risk if they're not properly educated on the potential hazards. So it's interesting because students do not fall under OSHA at the institution, but they do, certainly I think the school or college has a right and a responsibility to make sure that all people, employees and students, work safely. So this is a controversial issue, and I'd be happy to talk to anyone offline as well in more detail about maybe what the concerns are about student exposure. Employee exposure, certainly if there is something that's documented, would fall under the OSHA log 300 and would have to be recorded on the OSHA log. If it's something that requires or initiates maybe a loss of time or restricted duty, or if the person can't work with animals anymore, then that's an OSHA-related case that would have to be documented, and it's up to the institution or company to take the corrective action. So that would, the worst case would be involving human resources, reassigning someone to a different job, and so forth. Okay. Well, thanks everyone. I don't want to spend too much time. I do want to finish the presentation and get through some other questions you might have. Okay. So we did talk a little bit about ergonomics. So I know that I can't get feedback and a response. So the question here is, what is wrong with this picture? Is it ventilated? So I think it's pretty obvious from the picture, and I hope no one has something like this. But it is cages that are in a closet, not well ventilated. So it's, I guess, something that you don't want to have at your institution. Okay. I just want to touch briefly upon other types of contamination levels, kind of a concept that was developed by Boston University in terms of chemical contamination levels. there's a little deviation maybe from the main protocol, but these are potential risks to chemical agents. So a CC1, or chemical containment level, is a minimal potential harm to laboratory material. No additional measures, but certainly a review of the SDS would be important. A CC2 would be a moderate risk, so you might need to add additional measures like double gloving, secondary containment, use of a micro-isolator or ventilated cages, and low dust bedding might be helpful as well. And again, reviewing SDSs. A 3 would be serious risk, where you need maybe some type of double containment, a fume hood or a combination fume hood biosafety cabinet, a ducted system, and then additional secondary containment. And then certainly a 4 would be life-threatening, and then during any of this work, you would need to have kind of a very strict review of that procedure and protocol. So I don't know if anybody's thought of this before, but you could put this into the similar containment levels of the animal biosafety levels or the biological safety levels as well. Okay. The next slide is just an example of maybe some chemical agents and comments on how you would deal with those particular materials. So I'll leave that up to your individual locations to come up with some ideas on comments and restrictions. So I do want to talk about controls, and so the best way is to design a system where you have proper ventilation, substituting and eliminating the hazard if possible. But I know in research it's very difficult to eliminate the hazard entirely. Changing the way the work is done or by adding a tool might be helpful. Rotating workers more often to reduce ergonomic exposure times, chemical exposure times, and just basically helping with the occupational health program as well. And the last would be PPE is kind of the last resort. You should have all the others in place, but having respirators in place, gloves, hearing protection, and so forth. Okay. Before I forget, there is actually a organization in the Cambridge area where they do not wear PPE. It's a mouse facility, a small animal facility. And if anyone wants to know more about that facility, I'd be happy to share or put you in contact with the director or the veterinarian involved. But it's a new concept that if they have ventilated cages, they don't need to wear PPE, street clothes going in, but they do wear protective clothing when going into a BL2 area where there's animals. Okay. So engineering controls continued, certainly looking at the proper enclosure, dilution ventilation, looking at the cage design is important as well. Is it a well-designed HVAC system that can handle any particulates or exposure material? Are the cages designed with a filter top and ventilated racks? Okay, so all of these are very important. So the next picture is just an example of some filter top cages, some ventilated cage racks, and theoretically, if you have this system in place, you can work with multiple biological agents, multiple projects, although you may not want to, depending on your situation. Okay. The next slide is a basic biosafety cabinet that will filter out the air, remembering not to block your grills and working slowly in that environment when changing cages or working with animals, doing animal surgeries and procedures. Okay. Just a picture of someone using a cage changing station and wearing a respirator to protect against any exposures. Okay. The last picture here is on changes, cage changing stations. So when purchasing something, you want to take a look. I'm not advocating for this one, but to look at the design, look at the HEPA filters that would prevent any particulates, allergens, and other materials from passing through. Obviously, if you're using any chemicals in these stations, it needs to be ducted out of the facility and not have the air directed back into the room. Basic administration work. So as a summary, reduce your time with the animals, reduce the density of animals. You might say that you're going to work with 1,000 animals over a 3-year period, but certainly you want to also look at how many animals will be used per cage, maintaining good housekeeping practices in the room and with the animals, maybe using wet mopping in place of vacuuming or sweeping, and then make sure you minimize the release of any airborne materials. Okay. So I just want to get back to the questionnaire again. So when you're looking at kind of summarizing individuals, or should be reviewed at least annually. Are there any allergies that are seasonal that might be affecting their work? Are there any potentials for latex allergies? I know there's a controversy here. Latex gloves work better with animals, more dexterity, but there are latex gloves out there now that have a less of a risk when it comes to developing latex allergies. Most people will use nitrile gloves as well. And then look at, are people coughing, sneezing? Do they have runny noses? Is it related to their work in the lab with animals, or is it something outside of the lab? Keep all those masks. Do these symptoms go away, or do they remain in the facility? So I've talked off and on about the N95s and respirators. So this will reduce the inhalation and the hand-to-face spread of allergens. So again, think about whether this is something that should be required at your facility, which means you would have to have a respirator protection program in place for your workers. Okay. Some brief examples of success that I have seen in the field to reevaluate some of your work. This is an example of someone working with Staph aureus into devices in mice to study the way they minimize contamination for orthopedic hardware. It was originally classified as a BL2, requiring more time, effort, animal care staff handling, as well as additional PPE. After further review, the IACUC looked at the work. The staff is injected into the animal rather than spraying onto the fur and the outside surface. So this greatly reduced the risks, and the IBC approved it at the BL1 containment level. So it's always good to take a look at these practices. We always go to the other side and maybe err on the side of caution, but in some cases it may make sense to make some changes for protection, and it may or may not be driven by cost. Some institutions have undergone changes as well in their protocols and the assessments, so it's also good to take a look, maybe take some of the ideas from today, maybe incorporate them into your IACUC meetings, review philosophies, take a look at your SDSs, not MSDSs, SDSs, and look at the administration route, the dose, and the frequency, and any evidence from papers, scientific papers, of maybe things that have been improved over time. So here's one last case study just to keep in mind. I don't have a final answer on this, but something to think about. And we want to prevent this so that we can protect the worker and protect the animals from any problems. But a 21-year-old female worker at a pharmaceutical company developed symptoms, including hives on her forearms and hands. The worker began to suffer episodes of sneezing, nasal drainage, as well as watery eyes, chest tightness. She was transferred to another department where her symptoms changed. Okay? And so certainly we want to look at that potential exposure. Her symptoms ceased, I'm sorry. And the worker had a positive skin test to animal dander and to rat hair. Obviously, she was working with rats. She had elevated antibody levels in various rat proteins. So this is something that we want to prevent. We want to make sure that this individual had a family history of allergies, so maybe had a higher risks, no prior exposures to animals in the past. And this is something that maybe the questionnaire would have captured before she started working with animals. There may have been a recommendation to wear a respirator before working with animals. So unfortunately, in this case, the person could not go back to work with animals unless there was some additional controls put into place. And in summary, it's important to clearly articulate the policies and procedures. Okay? Make sure that you have an effective training program, and you would have to define what effective is at your institution or company. Implement the regulations. Facilitate research and teaching. Assure the public of quality care through your community member, and then determine strategies that would minimize the risks of and manage the risks as well. So I've been actually doing this for a very long time and doing safety for a long time, and it always goes back to proper communication. To me, communication is the key to any effective program, whether it's a biosafety program, IACUC related program, radiation program. So certainly keep that in mind, and hopefully you all agree with that as well. So I have some references. So I have obtained some of this information from Harvard, Boston University, UMass, Tufts, and others, and some case studies, and some information from my colleagues that I've known for many, many years. So with that, I want to thank all of you for attending the webinar. And James, I'll turn it over to you if you want to add any information, but you're welcome to contact me as well.
All right, thank you, Tony. And at this point, we'll open the floor to questions. We had a couple come in so far, and I just want to remind you how to submit a question. So instead of using the hand raise functionality, which won't work because you're on mute, we ask that you type in your question in the questions tab on your webinar panel, and we'll be able to field those for Tony. So as you're doing that, I just want to remind everyone that you will get a copy of this webinar tomorrow in an email. You'll also get the slide deck and a link to a short survey. So we'd greatly appreciate your feedback on the webinar and any ideas you have for future topics and improvement if you think there's room for improvement. Also, as a thank you for attending, we're offering a free biosafety program assessment or an initial assessment. Our EHS consultants like Tony are ready to give you help and advice when it comes to planning a successful biosafety program. We offer biosafety management planning, risk assessments, compliance reporting, regulatory training, and more. So to request that, I'm going to send a link in the chat box. Feel free to check that out, and you just have to submit a brief form, and we'll get you in touch with somebody that can help. And as Tony mentioned, his email address is there. I know he wouldn't mind getting an email from you if you have any questions later on. So Tony, let's take a look at some of the questions that have come in so far. We'll get to some of the early ones. All right. This is from Fred. Going back to respirator use, whether the employer supplies them or it's personal, and regardless of whether use is voluntary or required, use of a respirator in the workplace forces the employer to have a respiratory protection program, which includes training, fit testing, and medical surveillance. Is this accurate? I think he was getting some mixed signals there. Can you speak to that?
Oh, that is accurate. So you do need to have a respiratory protection program, whether it's voluntary or required. There are some things that you don't have to do if it's voluntary, but in all cases, I would recommend that you do training. You send people out for a medical clearance and have it reviewed, and they learn and know how to use the respiratory equipment and go through a fit testing. I think with the voluntary, I'd have to double check. They don't require that you do the training, but I would recommend that you do everything for the program. So I hope that clarifies the question. It might sound a little vague, but there is a responsibility that the company has, or institution, to provide employees with personal protective equipment, including respirators. So employees should not purchase their own. My recommendation is the employee goes through the EHS department or a primary contact to purchase these respiratory equipment so that the right program can be put into place.
Good advice. And Fred, if you have any follow-up questions, feel free to type those in or contact Tony directly. All right, so can you speak to the use of sharps in this context? How should needles and such be handled?
So certainly you want to use needles to eliminate the concern for needle sticks. So obviously all needles need to go into a sharps container directly, no recapping of needles. When handling needles with animals or injecting into animals, there needs to be a training program in place that has to be documented through your IACUC program and should be one of your policies. So in order to minimize needle sticks, really need to go through a training program with new people or existing people so you can go through those procedures. And again, as I said, never recap a needle. That's one thing that is under the bloodborne pathogen program as well as animal care and use programs.
Thanks. And can you walk us through, we talked a lot about risk assessments. Can you walk us through what a risk assessment or a good risk assessment would look like? Who's involved? Time period, frequency?
Sure. So we would want to look at the protocols in more detail. What is the experimental procedure for the use of animals? Taking a look at that first, and then looking at the different components that may create hazards. So we're looking at any potentials for use with biological agents, especially if they're highly infectious material. If it's select agent material, that's a whole different program with through the CDC. We'd want to look at the use of chemicals. Are the chemicals being injected? Are they being inhaled through a comb? Any radioactive material involved in that procedure. And how many animals? So there was a question about ergonomics. I'd want to look at how many animals are being used in this protocol and procedure. What's the husbandry involved? How often do you have to change the cages? Are the cages high or are they low? Where are they situated in the room? So I'd want to go through the details of that. I do use a checklist to go through that information in more detail so we don't miss anything. So that's something that, depending on the nature of the work and the facility, could take 30 minutes, could take a couple of hours, depending on the number of rooms or the number of projects that would be involved, and then follow up with some recommendations. So did I miss anything, James? Is there anything else in that, kind of walking you through that process?
Yeah, no, that sounds good to me. If anyone wants to learn more about it, again, get in touch with us. I'd say fill out that form or respond to my email tomorrow, and we can talk more in detail on that. Thanks for the overview. So getting back to sharps, I've got a question from Philip. He wants to know, do you have any recommendations for large animal workers when it comes to sharps? Some have stated that it's difficult to have sharps containers with them when injecting a horse, for example. I've seen several sharps-related injuries in this context.
So I can't ask Philip direct questions, but certainly large animals, I would recommend if you're working with sharps and potential for needle sticks or sharp objects, scalpels, things like that. Is the animal anesthetized? If not, if the animal is alive and walking around, then I would recommend having a second individual there to assist. Large animals like horses can also kick and bite. So during the course of injecting these animals, I want to make sure that they're calm and not putting you at risk, not only for the needle sticks, but also for other types of, blunt a kick or problem with that in that area. So that would be my initial assessment. At least get some additional help. Take a look at the needles that you have. Minimize the time that you're with the animal and calming down those animals. I've never been an animal worker, but we could take a closer look at the procedure and what's involved, how often that large animal is being handled, if there's any other animals in the room at the time. But certainly assistance, would not recommend anybody working alone during that procedure. Just a small story. I know we might be running out of time, but I had a student of mine years ago who shared a story about two people working with animals. One individual was holding a monkey. The other individual was actually getting ready to inject the monkey with a needle and syringe. And the monkey grabbed the needle and syringe and turned it around and injected the worker with the needle and syringe. So I don't think that would happen with a large animal like a horse or a cow, but certainly something to keep in mind and being very well aware of your surroundings and doing any of these procedures. So hopefully that answers the question.
Yeah, thanks for that. And I think we do have a time for one more. This is from Daniel. Do you know of any resources or procedures for evaluating the risk of chemical exposure to staff after an animal, such as a rat or a mouse, has been exposed to the chemical?
Good question. I want to know more about if there's any chemical that's excreted in the animal bedding, the nature of that chemical, going through a review of the safety data sheets, and then bringing that person to occupational safety to review any potential exposure. Not knowing the chemical, there may be some risks of certain types of exposures that would lead to respiratory concerns, and looking at the permissible exposure limits of that chemical.
Okay, and Daniel, if you're thinking of something specific, again, you can send that to Tony and he can give you some more advice there. That's all I've got for questions, so we will wrap it up. Thank you, Tony, for sharing your knowledge with us today. I hope everyone got some good takeaways that you can take back to your office, and we hope to see you on a future webinar. I'll give Tony a second here to give us any closing statements.
I want to thank everybody, and if you do have questions, feel free to reach out and ask questions.
Very good. Thanks, everyone. Have a great rest of the day, and we hope to see you on a future webinar. Take care.